pull down stuff. So you got your red stuff, which is what you're looking for. You got stuff that actually just came because it wanted to, right? And some of this other stuff came because the antibody was pulling stuff down that was not supposed to be pulled down. Then you clean it up, the proteinase can digest it. You reverse cross-link it. I forgot to tell you about reverse cross-linking too, because I forgot to tell you about cross-linking. I actually didn't forget, that was intentional. I didn't want to confuse you, okay? Basically, immunoprecipitation and washes, and then proteinase K digestion and reverse cross-linking. So now I've got just my DNA piece, okay? But my DNA piece has stuff. Now I make a library, I amplify this, I got my amplification. So everything gets amplified. So I have lots of red, I have some blues, I have some grays, I have some blacks, but I've got my reds enriched compared to the rest of it. Good, successful. Next generation sequencing, I talked about this. This is a paper we published several years ago, which has actually a video. I put it out there because it has a video of ChIP-seq. If you guys are interested, you can just go look at this paper, okay? And you'll know exactly how to ChIP-seq. This is all open access, you'll get it. Okay, similarly, cut and run, and you've got your sequencing done now, right? Cut and run, I'll come to this later, just because I don't want to confuse you, okay? Now I've got my thing. So the main problem in a computational analysis of ChIP-seq data is peak detection. Meaning, what is peak? Peak is just pile up. What is pile up? Pile up is just where your sequence was, right? So now, when plot the read counts against the genomic coordinates, the binding sites show a tall and pointy peak. So peaks are used to differ to places where protein binding or histone modification is there. Okay, very simple. And eventually you're trying to get a peak like this. That's your goal. So peak detection is the fundamental problem in an analysis of a ChIP-seq data or a cut and run data or anything like that, which is DNA binding. Okay? Okay. Now some of these slides are made by my postdocs. So I use this. I teach a bioinformatics course, and my postdocs actually go on and teach the computational analysis. So next set of slides are all by my postdocs, okay? So I must give credit to people where it's due because I'm lazy and I don't make my slides all the time. But postdocs also think technically, like you guys, so they put a lot of this stuff like that. Basically, peaks are regions where with reads clustered, so they can be detected from binned read counts. There's a lot of wording there, which you guys probably didn't get. How many of you got this? No? All right, then we're going to be next 30 hours is going to be torture for many of you. Counts from neighboring windows need to be combined to make an inference so it is more robust. And to combine counts, we use tools called max and sys whatever and whatever and whatever. But max is the word you must remember, because max is a tool made by a Harvard professor called Shirley Liu, which essentially is a peak detection tool. It's a biostatistics, bioinformatics tool that you can just go download and run. You don't need to understand what's inside of it. I'm going to try to explain a little bit of it, but if you get confused, because I do get confused too, I will admit, I'll be the first one to admit, don't worry about it. It's okay. It's just a computational tool. Tells you where the peaks are, okay? So I'm trying to come to a picture where I can explain to you peak detection. This is the data analysis that we do in the lab, okay? So we get the data from sequencing. ChIP-seq we did. I explained to you ChIP-seq. I explained to you, get your red counts. Red ones, remember the red ones? Right. You get data back in a file called FASTQ files. How many of you have heard of FASTQ files? None of you? One person. FASTQ file is just a file with all the data that came out of the sequencing. So it is essentially lines. If you imagine your Excel file, you imagine your columns, each row has a sequence in it, okay? Just imagine that to be a text file now. You can save an Excel file in a text format, right? A text file which has sequence after sequence after sequence after sequence after sequence is a FASTQ file, okay? because I have about 20 billion reads or 20 million reads that I got out of my data. Okay, so I actually sequence my library enough to be able to get at least 20 million reads per sample. 20 million is a number, don't worry about it, but it's in millions, or if not, several 20s or 30s or 50s or hundreds or 200s or whatever. Okay, why is that important? Because I need to have enough coverage to cover 6.6 billion bases, remember? Now let's think math. You guys are all math geniuses, I'm sure. You all got 90 plus marks in math. No? Yes? No? Ah, I know. I always got 100 in my math. I'm very proud of it. Okay, let's talk about this for a second. We have 3.3 billion reads. Got, right? Now we are in a genome sequence. What percent of that gets histone modified? 1%, 0.1%. The limit of detection is there, so you can't get everything out of it. Okay, so then you need some. Let's see, if you were to identify regions about, let's say, 0.1% or 0.01% of the genome that has H3K4 methylation in it, how many reads would you need? You don't know that. How many bases will you need is what I'm asking, not million reads. So you get a certain amount of data, okay, from your sequence. You want to have enough data so that you can cover that fraction at least 30, 40 times. Does that make sense? No. Okay. Let's say there are 100 bases in the genome. Let's think in terms of hundreds because it's easy to imagine, right? One of them gets modified. Okay. So I need, and how many reads will I need? I had 100 million bases. I need to have 1 million to be able to cover it one time. If I want to cover it 50 times, I need to have 50 million. Easy? So that fraction is what you think based on your knowledge. You think, okay, what percent of genome might have histone H3K4 methylation? And you say, okay, I want to cover it X number of times to be able to not make a mistake of losing it, right? That's how you calculate how many reads. So people by experience have figured out in the ChIP-seq experiments, normally with H3K4 methylation, you need 10 to 15 million reads to be able to cover it enough so you're not missing stuff. Okay, I mean, I can go more in detail. It needs to be in a range where you have saturated it, because if you don't saturate it, you're likely to find wrong information, and you compare two samples from each other because one may not have sequenced enough, other may have, and then you would get wrong answers in terms of comparison when you compare it. I don't think most of you got what I just said. People who got it are very smart or are my wavelength people, according to my numerologist, right? No, seriously. Okay. It's tough to explain, actually. I can explain it. It's just that I'm lazy. So, if you didn't have enough number of reads, right? By random chance, your reads in sample A may cover it, and in the sample B, it might miss it. So you need to have it covered enough number of times to be able to not miss in either one. That's what I'm saying. Does that make sense? Simplistically, okay? There's more computation to it, but that's what we do. We figure out what fraction of the genome is marked by whatever you're looking at. You sort of calculate a rough estimate of how much that should be and how many reads do I need to cover that portion. And then you say, okay, I need at least 20 to 30 times to not make an error of randomly sampling, okay? I hope that makes sense. If it doesn't, come to me later. I'll try to give you something to read. Okay. So we get a FASTQ file with a certain number of reads because sequencing is expensive, damn expensive. So you only want to get as many sequences as necessary to do this, right? FASTQ files. You first check the quality of the data you got from machine to make sure the machine didn't make an error, right? So that's called FastQC. You use a program called FastQC. It will tell you whether my sequencing was good quality or not. If it is trash, throw it away. Tell the sequencing guy, you take my money, give me my money back. He will say, no, you're not going to get it. You're going to have to go back and redo it. Then you go alignment. And somebody asked me, how do you align earlier, right? You align using a tool called Bowtie or another tool called BWA. And there are many more aligners now. Okay, so these computational people out there, that's what their job is. They go and make these methods and methods and methods. They improve on it and improve on it and improve on it. And then you have to keep changing your analysis after improvements. So people like me who are wet lab biologists hate computational biologists because they keep on making methods. In fact, we love them because they don't make methods. We won't be able to do anything, right? Okay, good. So it's a tool called Bowtie, okay? What a lovely name. You should go look into it. They use our BWA, it's called some Wheeler's algorithm, some B Wheeler's algorithm, BWA, that's more probably used these days. Every tool that you use, every computational tool, every analytical tool has some ups and downs, just like all our methods. They make those tools based on mathematical models, right? That, okay, what is the probability that this sequence aligns to this region and the next one to that region and the next region to, then like that, okay? So they use mathematics to design computational tools, right? What is the probability that this sequence that I gave you aligns only here? It's very simple, it should align, right? If A is A, T is T, C is C, 100% alignment may not happen, but you can then choose, you can tell it, when I will give me only those reads where 100% alignment happens, or you say, okay, give me those reads where there's only one error, or you can give it only those reads which have two errors and all, right? Most of the times we allow one mismatch for whatever it's worth, okay? I mean, people go on and do these repetitive processes and they figure out, okay, if I allow one, I get 90% true data and I can live with that. And people just use that, okay? That's how the world works. And then one person used it and then everybody else says, okay, this Harvard professor used it, so I can use it, and then on, right? That's what we do. We all do that, right? That's how science is built, by the community, not by one person. Yes, the Nobel Prize can be gotten by one person, but that Nobel Prize winner's work is actually built of many hundred professors' work who do publish a lot of papers, people learn from them and work together, but that Nobel Prize winning person actually made the discovery he made that actually changed the world, they think, but his discovery is just not made by himself, right? That's why it's a scientific community. Why I said to you earlier, we need many Joe scientists out there to work together to make drugs, okay? All right? I'll reinforce it all the time. We are selfless people. Are we? I boasted a lot about myself already a lot, I think, right? I'm not a selfless person. Most of us are selfless people. We go for our own, we want to be famous and this and that, but we are happy if our colleague gets famous too. And that day we are all working towards the same goal, making a missile, right? So Bowtie, you align your reads to the genome, to a reference genome. 50 years down the line from today, most likely you'll align the genes to that person's genome, because it'll be so cheap, maybe not 50 years, maybe 10 years or less. Okay, so I will have your sequence and I would have done ChIP-seq on your data and I would have aligned to that to say perfect matching. But reference genome is a genome, good enough to do what we want to do. Then you do peak calling using MAX, okay? Or you can do something called chromatin state, which I showed you earlier. I showed you a lot of that earlier, right? Those beautiful red and green and the things, peaks, those came from this algorithm called ChromeHMM. HMM stands for Hidden Markov Model. A friend of mine, Mr. Jason Ernst, he's in UCLA, he made the technique. When he was in my collaborator's space called Manolis Kellis, he's in MIT. I work closely with Manolis. And I work closely with Jason Ernst, and he's made this algorithm called ChromeHMM, Hidden Markov Model. How many of you have heard of this? None of you. It's a machine learning tool. I like to say machine learning, deep learning, because this is fancy stuff, guys. I didn't make it. I just use it, right? ChromeHMM. hidden Markov model. Please go read about hidden Markov model because it has implications way beyond what I'm teaching you. Hidden Markov model is an amazing model. Hidden Markov model discovered by somebody called Markov. I'm sure his last name was Markov. I don't know his first name, but I'm sure he was a Russian guy. You can look it up. Hidden Markov model. Hidden means it tells you hidden states from your data. Chrome-HMM, we inferred those chromatin states from our single histone data, right? Did you get it? No? No? We made HTK for trimethylation. We made HTK 27 acetylation. We made HTK for monomethylation. We fed all of them to the Chrome-HMM and said, now tell me combinatorial states that came out of that data. Which nucleosomes has combinations of these marks just based on the locations, right? So the Markov model then computed and said most likely hidden states are chromatin state 1, chromatin state 2, chromatin state 3, 4, 5, 6. I do this all the time in my lab. I use this tool all the time in my lab. When I get the chromatin state data, it just tells me, okay, these are the combinations, boss. Have you guys ever heard of combinations? Permutations? Combinations. It just gives me combinations. It doesn't give me permutations. Give me combinations. It says, these combinations occur. And then I go look at the data and I say, boom, boom, boom. This must mean there is an active enhancer here. Okay? And this active enhancer state is found on 50,000 genomic loci. So I now know 50,000 enhancers exist in this thing. And I mark them yellow, and that's how I showed you that in that beautiful picture. Where is the yellow? Get it? Some concept of it? Okay, and when I get H3K4 trimethyl and H3K4 K9 acetylation, I say this is an active promoter state, right? And I mark them into those red beautiful buildings, right? That's how I make my maps. That's my epigenome map, okay? This is how it happens. Magic. Well, that movie was brilliant, right? I didn't make it, I don't make it, but you see how you get there? By using beautiful antibody-based pull down. You pulled it down, you find your data, you go through these tools, and tools will spit out something. You annotate those states, and you say, here's my repressed state, because I know K27 trimethyl is my repressed mark. Here is my K9 trimethyl mark, is a repressed state. I'll show all of you this, by the way, later. But that's how I get to it, okay? I do peak calling. What does my peak calling mean? This, right? I get my max says, okay, here is a peak, okay? And I know exactly the coordinate of this because I know the sequence. I know the sequence and I mapped it to the genome. So I know it's on chromosome 1 at 10,500 location, right? And then I put it out there, boom, right? So my software puts the peak there. It says there are X number of piled reads here, and those piled reads should make this peak. Maybe there are 500 piled reads, okay? So the peak will be tall. Maybe there are 30 piled reads, so peak will be smaller. Okay, you get it? That's how peaks are made. So this peak here on the bottom here, which is not a peak actually, but looks like a peak, some pile up, that's maybe 50 reads. Maybe this is 500 reads, okay? That's how it makes. Cool? Boom, boom, boom. Okay, let's go back. I'm sorry, let's try to finish up in 4. Okay, so some of the key things in antibody when you do ChIP-seq is you want to make sure antibody is great quality. The less it binds to the non-specific things, the better data it's going to give you, right? Obviously, no questions. So people have done this kind of work where they've made a resource of good antibodies. But the problem with antibodies is the stuff I get from a rabbit after immunizing that is only so many mLs. I can get only so much antibody. The next time I immunize the next rabbit with the same antigen, I might not get the same quality antibody. So there is a batch-to-batch variation in antibodies, okay? I must know the problem with my experimental reagents. That's what a good experimentalist does. They know the problem with their experimental reagents. In this case, antibody is the biggest challenge. Get it? She was on the right side. I was telling her to explain the Western blot. This is a Western blot on the right side, right? So an antibody, gives you my beautiful one band here for Syn3B protein. So I know where my antibody is, right, where my protein is. But my second antibody might not be so specific, might pull down other stuff, right? So this antibody sucks on the right side. I can never use it. But antibody on the left side is a good antibody. I could use it, right? So the antibody flaws, it needs to be specific, as specific as possible to the protein of interest. Otherwise, you're going to pull down non-specific stuff. You're going to have lots of blue and gray regions, right? If you did that, you're going to have lots of these blues and reds, and you won't know what's my red and what's my blue. And if you, then you will, by mistakenly, wrongly label that H3K4 trimethyl is present where the blue stuff is, and it's actually not. And that might be at a gene where you might think, oh, this has a new cool gene that is expressed, but it's actually not. Unfortunately, that's a side effect, or what is it called? Off-target effect of your antibody. Okay, so you must know the problems with your thing. Cool. My tool cannot tell me the problem. I gotta know the problem. However, on that note, I can take care of something which is black. Can you guys guess how will I take care of the black stuff? Black stuff was something which had no antibody. So look at the black stuff. Okay. So black stuff is right here. It has no protein. Okay. It has no protein. So no nucleosomes essentially. It's just a nucleosome free region, but it still came along. Still came along. It liked to stick to the antibody. That's how we call it. How will I take care of it? I can do something called control samples. What does control in this mean? I can use my input. What is my input? My input is my whole cell extract. I can just sequence my extract. Okay? Without the antibody pull down. So I know which ones might actually just, or I could do a control, mock IP control, where I could use a non-specific, just a rabbit IgG. How many of you understood what I just said? None? Raise your hand if you did not understand me. Everybody should raise their hand. Good job. Okay. Okay. She explained how antibodies work, right? Antibodies have a specificity to certain things. Antibodies have this other region, right, which is just heavy region, right? There are antibodies that are just secondary antibodies. They just bind to random things. So if I use a control antibody, right? If some region of the DNA just binds to the antibodies for no reason, that will come out. That will be my, I can subtract that read out of my main experiment read to find where those reads came from. Does that make sense? Yes. Okay. So that's my mock IP control. Okay. So I use an antibody against a non-nuclear antigen, just like IgG, my secondary antibody or anything. Okay. So we normally will put some secondary antibody like a rabbit IgG. Boom. That's it. Okay. Or I can use my input control. So sometimes some parts of the DNA will just be easily amplifiable. Okay. That input control will take care of that because it's just input. Input means what I used at the end of the day for when I incubated with my antibody. Okay. So some regions. So that just controls for regions that are amplifiable. So this is real stuff. A lot of times you'll get things in your control like this. You would normally, if you didn't do a control, you'll call it a peak. But if you did the control, you'll not call it a peak. Okay, very simple. Always, a great scientist will tell you, an experiment is as good as its controls. If you don't do controls, the experiment has no meaning. If you go on to publish a paper and you didn't do the right control, the reviewer will trash your paper and will say, you didn't do the right control. There's no rigor in your experiment. It's called rigorous data science, rigorous science. Okay? Rigorous science. Remember that. Anything you do, don't cut corners. You understand what cutting corners mean? An experiment is as good as its control. Remember that word as long as you're a scientist. Remember that sentence. I tell this to my people all the time. An experiment is as good as its controls. Okay? Otherwise, I can't interpret the data. If I can't interpret the data, it's not interpretable. What did I tell you? My PhD advisor always said, one interpretable experiment, okay? A month is good enough. Maybe we'll get you a PhD. A week will get you a PhD faster, okay? All right, so we discussed sequencing depth earlier when I said you need to have 20 million reads or something like that. For a factor that looks like nice pointy peak like this, you don't need a lot of peaks. So you can do 10 million reads per replicate or something, okay? For a broad source factor like K27 trimethyl, remember I said they're like broad, they're always there. You need a lot more reads because they're broad. Of course, they cover a large amount of genome. So to cover that, you need large number of reads, right? Make sense? Make sense? Good. All right. I don't necessarily need to go to all details, okay? And these kind of, and this is like your, this is called lab, right? Your lab part. So this is of the GIAN. This is supposed to be a tutorial part of thing. What I'm doing right now, I'm teaching you and methods for an experiment. Other times I'm teaching you science. This time I'm teaching you methods. Okay, in my real experiment, when I go on, I give my people a FASTQ file and I ask them to analyze the data, and they analyze the data. You don't have a computer, I can't make you analyze the data. Okay, but I'm right now just telling you what experiments are needed. Here's the ChIP-seq analysis flow. So you do pre-processing, mapping, peak calling, peak finding. This is a first, we'll come to those things. I don't want to bother you right now too much. Okay, I'll show you some things that you can get some concepts out of this. Okay. I said you need to first check the quality of your peaks, right? You do this with this tool called FastQC. Okay. This looks like this. This per base sequence quality has a big red mark next to it. Means the sequencing failed. Okay. Whereas in the bottom here, this sequencing is a good quality sequencing, you see? Per base quality, meaning how confident are you that the sequencer did not make a mistake when sequencing that base, is what this QC matrix is, okay? You see all these are called box plots. You see the box plots going all the way, right? Like this. So all of this sequencing was not good. Has anybody done any kind of Sanger sequencing in your life? No? Okay. Then you guys won't understand enough of this. But nonetheless, there's a confidence that where A is, there is only A. Sometimes your Sanger sequencing will give you N because it doesn't know whether it's A, T, G, or C. So that's a bad quality sequencing. Okay. So in this particular example, in all of these positions, from 21, from let's say 13 to onwards, every single base actually has a problem. You see this? They're spread. It should be all around 30 for a good data. So you see a good data here. That's all around 30 or plus. Okay. A good data has a score of 30 or above. Fred quality scores, it is called. Okay. We do the alignment. I talked about alignment earlier. Burrows-Wheeler algorithm, that was it, BWA, okay? You use these other tools called Bowtie and all. They're different principles. They are different mathematics underlying. You can go look at it. And then you do a peak calling like this, what I just showed. So you first create a profile, you select the candidate sites, calculate significance of the enrichment of peaks over a control region, and you determine a cutoff threshold by a p-value FDR. Are you guys having biostatistics course? You understand what FDR means? Most of you know what is an FDR? No. False discovery rate. Most of you know what is a p-value? Huh? Probability value, yeah. Right, so you know a significance p-value. Significance is calculated by what is its chance of being real over a random occurrence, right? That's statistics for you guys. I'm not a statistics guru, so I'm not gonna get there. But p-value or FDR is simply the chance of your experimental observation being above random, right? Okay. I think you guys are all like, whatever, let me go. Are you? So here I told you earlier, you will get your sample and you'll get your input, right? Input is your control, sample is your real stuff. Here is a peak that you're trying to detect. So this is your reads. You aligned your reads and they look like this. You see how they look? Essentially there's a pile up, but there is some random pile up too, okay, from everything else, from your input. So essentially you are looking at probability of this peak being real over this random peaks, that random reads that are assigned. Does that make sense? That's what peak callers do. Yes. Oh, that's just a read 5 prime to 3 prime. You read, you sequence both here, this way and this way. I can get into details of that. You're randomly doing sonication, right? You're randomly aligning, as putting the ligation, right? So we are going to read from 5 prime to 3 prime, right? So some 5 prime to 3 prime will read from the forward end, some will read from the reverse end, forward versus reverse, and they will peak align at the same place, right? At the end of the day, sequence is same. Cool? No? Yes? Good? Confusions? Tomorrow, who's going to raise hands? I need to go through it again tomorrow? No? Okay. That's the peak that you'll be calling, okay? That's a significant peak. So Max is a great tool that uses the peak calling. I'm not going to go into this. Once you get the peaks, what do you get? Max, what does Max give you? Max gives you enriched regions. These regions in the genome are enriched for your antibody, right? Did we get it or no? So once you get the peak, that tells you chromosome 1, 10,500 to 10,700, where your peak is. That's the genomic region. Okay? Got it? Cool? Yes. And in genomic region, that region, I know what gene is near to it. Right? So now I can say, my gene locations, there are 23,000 genes. Here is a gene start site, and here is my peak. They are 200 base pair away. Okay? 500 base pair away inside the gene. So I can annotate the gene, which is nearest to my peak, right? Cool? Once I annotate that region, right, I can say, here is the enriched peak regions in the genome, and here are the annotated peaks that are near to that peak, annotated gene that is near to the peak. Okay, so I got my peak, I got my annotated gene next to it, right? So what I get? Well, I got enriched regions from Max, and Max then I assigned genes to each region. Okay, so I got my non-coding DNA that has a peak, my epigenetic mark on it, right? That's how I know epigenome is, right? And that's how I know which gene it regulates. Might be closest to that. So I take this, I visualize it in IGV. So I can take that file, I can put it in IGV that I showed you yesterday, and I can make that call that I just showed you earlier, those blue peaks, right? Then I say which gene is nearest to it. I say which genes are enriched over all my genes. That's called gene enrichment analysis. Have you guys heard of that? No? Okay. Gene enrichment analysis. Okay, let's say there are 500 peaks in a K4 trimethyl ChIP-seq, okay? And 500 peaks, let's say there are 500 genes next to it. Because K4 trimethyl happens on the start site, right? We should be able to do that. Of those 500 genes are 500 genes out of 23,000 genes, right? Of the 23,000 genes, somebody has gone out there and said, these set of genes are enriched in Wnt signaling pathway. These set of genes are Notch signaling pathway. These set of genes are enriched in metabolism. These set of genes are enriched in epigenetics. So I say, what is the ratio of my enriched genes over the genes that are in Wnt pathway over this? So then I say, okay, when there is a significant overlap, again, overlap above random chance, then I call enriched pathway. Get it? Okay. All right, everybody is sleepy. This is the last slide. Don't worry about it. We'll go tomorrow in the rest. Okay? Then I can do something called motif analysis. So I have the DNA sequence for enriched regions. I can say, is there a specific DNA sequence that is enriched in the enriched regions? Does that make sense? So the sequence, right? So I can say ATGCCG is always enriched in these. And that is a transcription factor binding site for MYC or for AP1 or for TCF4 or for X, Y, and Z. So now I know which transcription factors may be actually enriched, may be binding to those places. And then I know what is my biology. I know my signaling pathway. I know Wnt signaling. I know TCF3 is enriched, and I know TCF3 is downstream of Wnt. So I know Wnt signaling pathway genes are enriched because TCF3 is regulating this through modification X that I was analyzing to begin with in ChIP-seq. Get it? That's how you make inference of the data. And you say, boom, I know now how this works. My cancer is caused by epigenetic regulation of Wnt pathway. Did you get it? It's okay. That's the conclusion I made out of my unbiased discovery. Okay? And I might have seen, oh, LIF pathway is enriched in MPNST, the cancer I told you about earlier. And I said, boom, MPNST may be caused by LIF pathway. And there is an inhibitor to the LIF pathway. So let me go treat my cancer with that LIF pathway inhibitor. Okay? Boom, I connected the dots. I'm a scientist. I'm a detective, right? Cool? Gene set enrichment pathway tools. There are tools called ChIP-seeker, Enricher. You can use them. And that's it. I'm not going to do the lab with you, so I'm not going to go into this. But I'll send you the slides. After this, there are all the tools. So this is what we're talking about. Here are the codes. You can use these code to actually download the software. And actually, if you actually had a BAM FASTQ file, you'll be able to run it and come to what I just showed you. So there is all of these commands here, okay? This is what I run through the PhD students in my company, in my lab when I do this, okay? We'll not worry about any of these. We'll get to this tomorrow. This is it. I like to end here. You'll get these maps with those beautiful things, and you'll be very happy that I made an epigenome map from my patient sample data X, okay? Good. Have a good night, guys. Everybody's sleepy right now. Have you ever taken an intense course like this? I never have. Okay? Good. Enjoy, guys. Thank you for dealing with me. That's not funny. Okay. Are you guys having fun? Is this Giri good working?